TBIM Day 2. We have a lot of great talks for y'all today, and we have also an invited speaker after lunch today, so hopefully everyone sticks around for that. So to get started, our first speaker is Donovan Salazar talking about the effects of hyposalinity and high nitrogen on the red seaweed gracilaria.
Thank you. Good morning. How are we doing today? All right, cool. So today we're going to be discussing the effects of hyposalinity and high nutrient spikes on the red seaweed Gracilaria chickavahiae and some of the implications for water quality in the lower Laguna Madre specifically. Is the clicker on? Okay. So the Arroyo Colorado watershed in South Texas drains into the Laguna Madre, which is introducing a lot of fresh water and nitrogen, nutrients and phosphorus as well. But in this experiment, we focused on nitrogen. So the lagoon is characterized by having a higher than average salt concentration, about 40 parts per thousand. So in extreme weather events such as Hurricane Alex depicted here, there's a lot of fresh water flowing into these waterways and across the Arroyo Colorado watershed. And it has been recorded depressing desalinity in the lagoon for like long periods of time. So as you can see here in the shaded portion of the graph, this represents the salinity in the lagoon across these various transects, and that red circle there, that's the mouth of the Arroyo Colorado, and that corresponds to the red. I don't know if this. Yeah, there we go. So that corresponds to these red arrows, and you can see from July 2nd, 2010 to August 6th, and even to September 25th, the salinity in this location is very low. So over the 60-day average, it did not go above 10 parts per thousand. So that's really bad for an area that's used to having, higher than average salt concentrations. So with all this fresh water, a large amount of nutrients is transported across the watershed. Over 350,000 people live within the bounds of the watershed that create sewage that will eventually end up as nutrient-rich wastewater that is going to flow into the Laguna Madre. So this nutrient concentration is further increased during precipitation events, mostly due to runoff. So there's a lot of agricultural land across South Texas, right? So we got cotton, citrus, sorghum, a lot of cash crops, right? A lot of these cash crops use fertilizer that are, obviously very high in nitrogen and nutrients. So these eutrophic conditions, along with the large amount of fresh water, can have a substantial impact on coastal waters. And there's a number of ecological ramifications that come with this freshwater inflow and nutrients. So the most extensively recorded have been lower salinities, obviously nutrient input, increase in organic matter, pollutant input, and in overall degradation in water quality. So what does this mean for marine plants? The effects of the previously listed ecosystem changes have been recorded negatively impacting the overall health of seaweed and other marine plants. I focused on seaweed in my study. To measure the health of seaweed, previous studies have monitored the photosynthetic, respiration, and growth rate, as well as the chlorophyll concentration within the different marine plants. So all of these I studied in my experiment. But I focused on the impact that freshwater input and nitrogen specifically had on the health of the red seaweed, Gracilaria chickavahiae. I did this because it's a very common seaweed and it's euryhaline, so that means it's able to survive within a range of salinities. It's also one of the most effective absorbers of nitrogen out of all the red seaweed species. So the purpose of this study is, again, to monitor it's simulating the precipitation event very similar to what we saw in Alex in the earlier slides. So we're going to decrease the salinity over a period of time and introduce nutrient spikes. So after this, throughout the duration of the experiment, we were monitoring the growth, photosynthetic rate, respiration rate, and chlorophyll A concentrations. The literature states that the species is able to survive a wide range of nutrients, but there is a limit to how much it can absorb. Gracilaria has actually been used in bioremediation efforts across, across the states, but not so here in Texas, not as of yet. So to monitor the photosynthetic and respiration rates, I use the light-dark bottle method. So we measure the initial O2 concentrations in these closed system bottles, introduce seaweed, and measure the O2 concentrations after an incubation period of 2 hours. And the difference is the rate at which the seaweed either respired or photosynthesized. Okay, so I did two experiments for logistic reasons where the two separate experiments had a control group, hyposalinity group, combination nutrient and hyposalinity group, and a nutrient group. So the only difference between the two was the type of nitrogen that I introduced. I introduced 120 micromoles of ammonia in experiment 2 and 120 micromoles of nitrate in experiment 1. So this is following the experimental design. In experiment 1, again, nitrate. Experiment 2, ammonia. So after a brief acclimation period, I decreased the salinity from 35 parts per thousand, which is like average seawater, down to 0 parts per thousand. So we did 4 salinity decreases on day 1 from 35 to 25. 25 is still within Gracilaria's habitable range, by the way. It's going to be important for later. And then from 25 to 15, 15 to 5, and after each salinity drop, we added a nitrogen spike. So, you're not going to add nitrogen before changing the salinity. Doesn't make any sense. Okay, so to the meat and potatoes of this. Over the course of the experiment, all of these seaweed treatments did grow. However, in experiment 1, groups experiencing hyposalinity grew significantly less than that of the nutrient and the control group. In experiment 2, only the control group grew significantly more than its counterparts. Okay, so this is the gross primary production of the nitrate group, experiment 1. You can see in the upper right-hand corner, this is the salinity key, 35 parts per thousand. Here, throughout the duration of the experiment, the control and the nitrate group did not experience any decrease in salinity. Here, we dropped the salinity, and you can already kind of see a pattern starting to emerge. You can see a clear decline in the net primary production, or the photosynthetic rate, of the groups experiencing hyposalinity. The red is the respiration rate. So right here on day 7, you can barely see any red, right? That means the seaweed is no longer respiring as effectively as it was compared to the other treatments or even compared to day 1. So what does this mean? That means the seaweed is experiencing a significant amount of stress and it's not having a good time, basically. We can see a very similar pattern here with the ammonia group. Again, very same structure. But on day 1 to day 3, this is 25 parts per thousand, still within the Gracilaria's habitable range. We can see it increase in its photosynthetic rate. The respiration does decrease, however, for the hyposalinity group. And again, by day 5 and day 7, almost no respiration. In fact, I got some negative numbers in my experiment, but averages and whatnot. Here in the control, it actually increased from day 1 to day 3 significantly, but then maintained consistent over the rest of the experiment. The ammonia group decreased significantly from day 1 to day 3, but again, leveled off as the experiment progressed. So why might there be a significant decrease in photosynthesis? So we looked at the chlorophyll A concentrations of each of these treatments. So this is the nitrate experiment 1 and you can see there's significantly lower chlorophyll concentrations in those groups experiencing hyposalinity. So this is the control, this is the nutrient group. And we're seeing something similar here in the ammonia treatment. However, the chlorophyll concentration was only significantly lower for the group experiencing hyposalinity. So what does this mean for the Laguna Madre and other coastal ecosystems? Well, it appears that over extended periods of time, the changes in salinity is going to have the greatest impact on the gross primary production and overall health of the red seaweed, Gracilaria tikvahiae. The addition of nutrients does not seem to have a significant impact, either as like a buffer or as an additional stressor on the seaweed. So it's not really a stretch to assume that a species as hardy as Gracilaria, hardy is a kind of a loose term, but resilient is something that's more like accurate. This might be happening at greater rates, a decrease in health to other species, something like less resilient. So as freshwater is periodically introduced to the arroyo or from the arroyo into the Laguna Madre, we'd likely see an increase in dead seaweed in large precipitation events. So this is an algal mat we found right off the coast, right off the mouth of the Laguna Madre. That is a lot of seaweed, right? So if all this were to die, this could likely lead to further nutrient pollution, smothering of seagrass, a degradation of water quality in the form of like hypoxia. And not only will this large precipitation event like lower water quality by introducing nutrients, pollutants, and other runoff, but it will increase the water quality over a longer period of time as different marine plants and maybe even benthic organisms are not able to withstand the changes in the ecosystem. So future studies may benefit from determining how these conditions progress and further degrade the coastal ecosystems. Thank you.
Oh, you stay up here. So we have a lot of time for questions. You did a really good job on time. So anyone have any questions for Donovan?
Here, practice.
Okay. Good. Okay, everybody, please welcome Nora Bleth to the stage. And she's going to be talking about Baffin Bay and looking at microbial source tracking, examining non-point sources of fecal pollution.
Good morning, everyone. My name is Nora. I am from the Turner Lab at TAMUCC. I'm a PhD student, and this is part of my PhD research. How many of you guys have been out to Baffin Bay? Oh my goodness, so many of you. That's so great. The next question, if you guys didn't raise your hands for that, was going to be, do you guys like bays? But I mean, it's in the title of the conference. All right, so I'm going to be talking about fecal pollution. But first, I want to talk a little bit about Baffin Bay and what it does for South Texas. It has been known as the jewel of the Texas coast. It's got great hiking, fishing, boating, hunting, birding, wildflowers, and habitat for lots of critters and ecosystems. Just take a look at some of those pictures over there. Those are taken during my project. Aren't they gorgeous? However, the geography of Baffin Bay and the geography around Baffin Bay make it intrinsically vulnerable to fecal pollution. So just a quick orientation to the map. Here's the bay right here. It is enclosed by Padre Island right here. So that's going to lead to minimal tidal flushing and long residence times. The water's staying in there for quite a while. It is also hypersaline, so extra salty, and the land around it is mixed use. It's used for a lot of different things. This map here shows the difference between ranch land and farmland. So over here by one of the inflows, this is Petronila Creek right here, is a lot of farmland. It's mixed more by San Fernando Creek. Up here we've got Kingsville, so some population, people, housing. And then over by Los Olmos Creek, it is mostly ranch land. And this has consequences on the water quality of the bay. There has been a rapid decline in water quality over the past couple decades, from algal blooms to hypoxia, fish kills, and the impairment of 3 major tributaries due to high fecal indicator bacteria pollutions. Those 3 tributaries are Petronila, San Fernando, and Los Olmos, and that is what I'm going to focus on for the rest of this talk, is the fecal bacterial pollution. So fecal pollution, like any other pollution, operates in point source pollution, where you've got a single identifiable source like a industry, a factory, some kind of industrial runoff. You can figure out exactly where that source is. And there's also non-point source pollution, where there's many unidentifiable sources accumulating in the water. So in this picture here, we've got some agriculture, fecal pollution, the cows are pooping, pig is pooping. We've got some urban, we've got a little city dog there, and some people, some wastewater. So because of that non-point source pollution, there can be many, many drivers to what's happening, particularly in Baffin. So let's go over some of the drivers, things that cause fecal pollution, such as bird loitering, aging infrastructure and septic systems, pet waste, urbanization, population growth, sea level rise, stormwater runoff, and livestock. So just taking a look at a couple of these, they could all be happening in Baffin, actually quite a few of them, like right now. So we have to figure out what's going on and identify these sources and drivers so we can manage them. So this is where my experiment comes in. We did monthly water sampling in the watershed. So right here, these little green dots, those are all my sites. There's 6 in the bay. There's 3 in San Fernando Creek, 2 in Petronila Creek right here, and 1 in Los Olmos Creek. We did this for 18 months, so we had 18 sampling events at 12 sites for a total of 216 water samples. And we divided it up into those 4 systems of Baffin Bay, San Fernando, and Los Olmos Creek. And a quick note here, the red means impaired. At the beginning of this experiment, only San Fernando and Petronila Creek were impaired. Los Olmos was added to the list of impaired creeks in 2024. Forgot Petronila. So the first thing we did was the traditional fecal indicator bacteria. This is approved by the EPA. It's standard across the board. This is what you would look at to check the fecal pollution at any beach or something like that. So we go out, collect the water samples. There they are. There's me. There's my lab mate Bella. Here's some of our water samples. And then we do a test called Enter Alert on marine water or Colert on fresh water. We take this little packet here. It's got bacteria food in there. If the bacteria are what we're looking for, that fecal bacteria, they will eat it, they will metabolize it, and then they will glow in the dark. And the ones that glow in the dark, you count the wells and you calculate something called a most probable number, which is just how many bacteria are in 100 milliliters of water. However, as useful as this is, it is not source specific. We cannot tell from this tray where the fecal bacteria are coming from. It's like trying to solve a mystery and you find fingerprints at the crime scene, but you can't figure out who they came from. And it's also prone to false positives in saline environments. This is really problematic, especially in a place like Baffin, where it's not only hypersaline, but the tributaries have salinity gradients along the river. So there's some suspicion that these tests might be having false positives. And I will talk a little bit about that more when I show you the data. Here's what I got. So in Baffin Bay and Los Olmos, we used Enter Alert because it's a marine system. And in San Fernando and Petronila, we use the Colert E. coli because it's a freshwater system. And we look at something called a statistical threshold value. This is set by the EPA. It is that no more than 10% of samples can exceed this value in a given time period. So for enterococci, that's 110 MPN. And for E. coli, that's 320 MPN. So no more than 10% of samples can exceed this. Well, when it came to the enterococci in Baffin and Los Olmos, 17.24% of our samples were exceeding that number. And within the tributaries, Petronila and San Fernando, 80% of them exceeded that number. So we need to look a little closer. We have to do something to figure out where this is coming from. And so we can manage those individual sources, because all we know right now is that it's there. So we do something called microbial source tracking. This is the meat of what my lab does. First, we go out, we collect the water, we filter it through this vacuum apparatus right here. This is my lab, actually. And then we extract the DNA from the water samples. So all that water sample, after it's filtered, ends up on a little piece of paper. We cut it up, use a little kit, extract the DNA, and it gets in a tube. And then we do droplet digital PCR. Droplet digital PCR is basically we have the sample in here and we have the PCR liquid in here, and then we use an oil and it partitions it into 20,000 tiny little droplets, and each of those droplets is a PCR reaction similar to the Colert test. It will fluoresce if it's positive for the gene in question. So we can use genes associated with individual hosts to determine how much that host is pooping, which means we have to examine the potential poopers in Baffin Bay. So we look at the land use around there, we look at the habitat, and we think, who could be pooping? Well, it could be people. There's an estimated 11,000 septic systems, some of which are getting older in that watershed. It could be shorebirds. There's lots of birds flying around. People go there for birding. It's beautiful. But there are also some landfills, and birds like to eat trash. There are 100,000 head of cattle in that area. It's big for ranch land. And then there are also the invasive feral hogs. So we got host-associated markers, primers for each of these, and we thought, let's see how much each of these is pooping. Okay, so this is the PCA. I'm not going to spend too much time on this. What I want you to note is that the samples cluster by site. Baffin Bay is this huge watershed that spans multiple counties. It warrants a holistic investigation, but because of this site clustering, it gave us justification to examine the individual characteristics of each system within the whole. So I'm going to go through this graph. We're going to see it 4 times, one for each marker. So right over here in the box plot, we have the system on the x-axis. So Baffin Bay, Los Olmos, Petronila, and San Fernando. We have the concentration of the gene marker that we're looking for. In this case, it's the human marker, and it's logged. So every step of a whole number, so from 2 to 3, that's a jump of a 10-fold increase. And then over here, we have a spatial interpolation. So it's basically a heat map. We've got our sites, they're all listed here. The redder the color, the higher the concentration. So let's take a look at this human marker. These sites by San Fernando, right by the highway, this is right where Kingsville is. San Fernando is testing much higher for that human marker than the other sites. For the gall marker, it really lights up. We've got lots of orange, lots of red, but this is more in the Bay and in Los Olmos than it is in San Fernando and Petronila. So take a look here. There's Bay Site 1, Bay Site 5. I think of these as like the arms of Baffin Bay. I don't know. To me, it's like the 3 arms with the 3 tributaries leading in. But it's not so high in Petronila and San Fernando in the northern parts. And then we take a look at the pig marker. Once again, this is higher in Petronila and San Fernando, which makes sense because pigs are land animals and they can't go poop out here in the middle of the water. But San Fernando is leading again. And lastly, this is the cow marker. This is the highest marker overall. This is one of the main drivers. We see bright orange in San Fernando, little less in Petronila. If we remember that land use map, Petronila had more cropland than San Fernando did, but it is omnipresent throughout the entire area. Highest in San Fernando, but omnipresent. So I'm going to take a look at some summary stats and tell you a little story here. So first, I want you to see the percent detected. This is how much, for each sample out of our whole sample set, the percentage where we detected that marker. So for Baffin, we detected the human marker in 9% of samples. We detected the cow marker in about 52% of samples. Los Olmos, 22% of samples we had human marker. 83, we saw the cow marker detected. Similar with Petronilla, 44, 91% of samples had that cow marker. And then in San Fernando, we had 75% of samples test positive for the human marker, which tracks. There's a city nearby, there's more people. But 98% of samples had the cow marker in it. Now let's take a little bit of time to look at the difference between San Fernando and Petronilla Creek. When we saw that fecal indicator bacteria chart, Petronilla had a higher median fecal indicator bacteria than San Fernando. Well, if we take a look at these concentrations for every single marker, San Fernando has a higher concentration, higher median concentration than Petronilla. Why could this be? Well, my lab hypothesizes at the moment that it has to do with Petronilla's salinity gradient. It tests more saline than San Fernando Creek, but they were both tested with the E. coli test, which is meant for freshwater. Well, as we saw earlier, higher salinity means more false positives. So we suspect that those fecal indicator bacteria tests are projecting more false positives. And what we're actually seeing is a remnant of the salinity and not a remnant of what is actually there. So San Fernando Creek seems like the place we've got to target. And lastly, to further hone in on that point, San Fernando Creek had 3 of the maximum values for every single marker. It had the max human, it had the maximum pig concentration, the maximum cow concentration. The only one that it didn't have the max of was the gull, which happened in Baffin. So if we put it all together, it looks like this. It's all of those bar graphs I showed you earlier mushed together. We've got Baffin Bay, Los Olmos, Petronilla, San Fernando. Each marker is a different color. Human in blue, gull in green, pig in pink, and cow in yellow. The gene copy concentration is along the y-axis. Human fecal pollution is not driving the impairment by fecal indicator bacteria of Baffin Bay and its tributaries. It is cattle and it is gulls. Pigs contribute a fair amount, but cattle and gull are driving the fecal impairment of Baffin Bay. So here's the takeaways that I want you guys to walk out of here with. First is that we do have those high exceedances in fecal indicator bacteria. Because it is the standard test, it does its job, it's done its job for a while, it is important to note that that is happening. It is exceeding the standard. But when it comes to management, we have to look and see that it is primarily driven by cattle and gulls. And so those need to be our target species for managing fecal pollution. The second is that San Fernando is the hotspot. Even though Petronilla tests higher, we should probably be testing it with the enter alert test and not the Kohler test because of that salinity gradient. San Fernando is where we need to target because it's the hotspot for human, cattle, and pig fecal pollution. And lastly, I want to leave you guys with that fecal pollution is complex. It's got many drivers, it's got many sources, and we need more complex studies that utilize microbial source tracking to better understand these dynamics. Where is this going? Well, we're going to do a human risk assessment on Baffin, and then we're going to work with stakeholders to inform the Baffin Bay Watershed Protection Plan. And what's coming up next, hopefully I'll be here next year presenting about antimicrobial resistance in the Bay. Thanks to the lab, everyone who helped on this project. It's a huge effort, and I'll take any questions.
Time for at least one question. So does anyone have a question for Nora?
Yes. They were surface level samples, just like a grab sample. I don't have the distance off the top of my head, but San Fernando and Petronilla have small towns around them in Kingsville, and I know that I think there's 3 within that watershed area.
Okay, please welcome our next speaker, Katie Lloyd, to the stage. She will be talking about the bioavailability of photo-bleach dissolved organic carbon from environmentally prevalent plastic nurdles in coastal waters.
Hello, everyone. Thank you for that introduction. As she said, I'm Katie. Um, Excited to talk to y'all this morning so I can get done with this and stop being stressed out about it. Um, so, yeah, there's the gist. Getting right into it, we know plastics are really, really bad. The estimate a few years ago was 400 million tons of plastic produced worldwide just in 1 year. And the estimate for how much of that enters the ocean per year is 4.8 to 12.7 million tons, which is also not good. Um, Once that plastic enters the ocean, it goes through various weathering processes such as UV degradation and oxidation, as well as thermal, mechanical, and biological forces. During those weathering processes, it is fragmented and broken down into microplastics and nanoplastics. And it is also broken down into leachate, these really small compounds that just sort of go off into the water. And one of those that I'll be talking about a lot today is dissolved organic carbon. Yeah, so once those microplastics and nanoplastics are broken down, they're ingested and taken in by organisms. They're going into plants, soil, fish, basically everything that you can think of in the ocean, they're there. And they've also been found to bioaccumulate as they go up through the trophic levels. So this poor shark is probably going to have a lot of microplastics from that fish who ate a lot of microplastics from zooplankton. Yeah, and then they also produce harmful chemicals when they're broken down. So you have bisphenol A and a whole bunch of other ones that are going to negatively impact these organisms, reducing their growth rates, their reproduction, and their lifespans. So I talked about some of those weathering processes. The two that I'm focusing on in my project are photodegradation and biodegradation. Photodegradation is the breakdown of plastics with UV irradiation. There's a lot of different mechanisms that sort of do that when it's getting irradiated. So you have chain fission, the alteration of the shape of the molecules, et cetera, et cetera. And during that process, low molecular weight compounds and DOC are also produced. And then you have biodegradation, which is the assimilation and digestion of these microplastics by microorganisms to support their metabolic activity. These rates of degradation are pretty low typically because plastics are hard to degrade. They just have high crystallinity, a lot of strong carbon bonds. They're hydrophobic, so these microbes have a hard time with that. But UV pretreatment does help, and together these processes can work together to degrade these plastics and maybe get rid of them eventually over hundreds of years. But we know all this. We know that they work together really well. We know that UV degradation is a great precursor for biodegradation. But what we don't know too much about is the products of those plastics, the DOC. How is that biodegraded? How is that assimilated after it has been produced during photodegradation? So that's what I'm focusing on, trying to look at those chemical properties and the bioassimilation of that dissolved organic carbon. So my objectives are to determine the UV sensitivity and photodegradation potential of 4 common polymers, evaluate the bioavailability of microplastic weathering products and DOC to understand the longevity and role in marine ecosystems, and then discover which of my microorganisms most effectively utilize plastic DOC with the hope that we can use that information for remediation and engineering of those enzymes that degrade the plastic in the future. So my plastics of focus were high density polyethylene, polyethylene terephthalate, polypropylene, and polystyrene. These are just some of the most abundant plastics globally and locally. They have a lot of different characteristics that make them interesting to study. RPP and PE are aliphatic compounds. They have very simple chain structures. On the other hand, PET and polystyrene have more complex aromatic rings, maybe more susceptible to photoradiation because of those aromatic chromophores. There's a lot of things that you can sort of guess is going to happen with the degradation of these plastics, and it doesn't always go the way that you think it's going to go with those chemical structures, which we'll see. But and then I focus on nurdles because we're in Texas and they're everywhere. There's hundreds of thousands of millions of them all along our beaches because of those factories. So keeping it local, keeping it relevant. So first bit, photodegradation. Try to go through these methods pretty quick, but we put our nurdles in artificial seawater in this solar simulator. It simulates noon summer sun in Port Aransas. So we did that for 4 and 8 week time periods. That 4 week period is equivalent to about 3/4 of a year, that noon summer sun, and then the 8 weeks is 1.5 years. Then we separated those samples into the photoirradiated nurdles and the irradiated DOC leachate. We analyzed the photoirradiated nurdle weathering and oxidation with FTIR and SEM imaging. And then thank you to Ryan in the core lab for some of that TOC analysis of the DOC that was produced from photodegradation. So this is the first result, our FTIR. I just want to point out a few things about this. So we chose 4 different bonds that tend to see a lot of changes during photoirradiation. The most significant one here is this carbonyl bond. You can see that top line, the darker one, is the pre-irradiated nurdles, and then that bottom one, lighter blue, is the 8-week irradiated nurdles. The transmission percent decreased, which means that there are more bonds in that structure over that time period, and there's more oxygen within the structure that's allowing those carbon oxygen bonds to be created. And then this is just an easier visual representation. Those indices, those bonds graphed, and you can see that there's a pretty significant oxidation over that time period. And then these are our nurdles after 4 weeks of irradiation. You can see the polystyrene ones have some yellowing, which is evidence of photooxidation, degradation. And then this is our polyethylene SEM imaging. After 4 weeks and 8 weeks, there's cracks and wrinkles, just some evidence that degradation was occurring. And the most important thing that I want y'all to take away from this is that it is very important when you're looking at photooxidation and degradation to analyze weathering with different methods and instruments, because you might see some different results and you need to put the pieces of the puzzle together. So this is our PET oxidation. You can see it looks like oxidation actually went down, and then you can see there are a lot of cracks and fractures formed in that PET nurdle. So signs of photodegradation on one hand and limited signs of oxidation on the other hand. Don't really know why. We're going to try to figure that out. But yeah, a lot of different things. And then this is our DOC produced after 4 and 8 weeks of irradiation. You can see that polypropylene produced the most DOC. Polystyrene and PET were pretty low, and that might be because of dissolved oxygen limitation in our samples, or because of some sort of physical or chemical characteristics that prevent them from being oxidized, which was against my hypothesis initially. So this looks like a lot of DOC, and it is in the context of the DOC in the ocean, because those concentrations are relatively low, and this means that this plastic is providing a lot of organic carbon to the ocean once it is fully degraded like this. But it is still only about 1.5% of the mass of the nurdles that we irradiated, which means it's going to take a very long time for these plastics to be broken down. Hundreds of years. I think I calculated like 400 for 1 gram of these nurdles. For the biodegradation, I'll keep it quick. So we took the DOC leachate from photoirradiation, inoculated it with artificial seawater and natural seawater for our microbial consorta. That was in there for 35 days. Then again, thanks to Ryan for DOC analysis, basically looking at the acquisition of the DOC by the microbes. And then I measured microbial abundance with flow cytometry, and the community data we sent off somewhere, and they gave us our data back, and I just got that back last week, so very preliminary. I haven't gone through all of it, but yeah. So this is our DOC acquisition. You can see a significant decrease of the DOC observed, which means it was being assimilated or mineralized by those microbes in the tubes. The bioavailability percentage was about 75 to 95% for all of the polymers. They were not significantly different. But yeah, that just means a lot of the DOC is actually being used by the microbes, and they can use that, which is much better than the plastics because they can't use that much of that. And this is our first order decay model equation for that. I won't go into that right now for the sake of time. Boop. So this is the DOC acquisition again on, there it is, the left side, just without the first order decay fit. And then on the right side over here is our change in biomass over time. The main point of this is the DOC continued to be assimilated during that time, even as we saw a decrease in the population of our microbes for 3 of our polymers. PET increased pretty significantly, but we don't know why yet. So our assumption is basically that only a small subset of the microbial population effectively mineralizes the DOC and survives. So I wanted to check that with the microbial data that we ended up getting this past week. I got overwhelmed and didn't do all of that. But this is at the class level, so definitely not what I want. I want to go to the genus and species, but I just tried to get some sort of idea from this data. So some of the conclusions were that the composition differs among the same treatment over the course of incubation. Where are you? Yeah. So this PE1 and PE2 vial at day 21 had significantly different compositions, even though it's the same leachate type and same day. And then these communities were mostly dominated by gamma and alpha proteobacteria, which are the blue and orange sections. I was looking for an increase in that in microorganisms for PET at day 21, about right here, when the population spiked. There was a large increase in alpha proteobacteria and beta proteobacteria, but these aren't unique to that one vial, so I don't know if that explains the increase in the population or not. And I don't know if it has something to do with PET itself. Maybe it's just more conducive for bacterial growth because of its structure, more biolabile. Don't know that yet, but yeah, so the main takeaways are that photoradiation significantly changes the surface chemistry of polymers, particularly polyethylene and polypropylene, leading to increased oxidation and production of significant dissolved organic carbon. DOC from the photoirradiated nurdles, regardless of polymer type, is quite bioavailable. There's proof of full DOC degradation, not plastic, which helps explain why so much of plastic mass is unaccounted for, because if it gets broken down in the DOC, completely used by microbes, it's gone. So we can't track it. And I don't know if it's inhibitory yet, because the blank vials had actually higher microbial abundance than a lot of these polymer vials, which is maybe the connection to water quality, since I feel like it was loosely connected to this session. But this plastic can be have negative effects on microorganisms, but they can also use it. And then DOC acquisition may only be done by a handful of microorganisms from the microbial consortia. So my next steps are to obviously dive deeper into my 16S data. My DOC, I just did LCMS, or Ryan helped me with LCMS, and hopefully we'll get that data and get some more chemical understanding of that DOC. I'm gonna make some cool graphs and actually use R, because I've been using Excel this whole time because I hate R. And then I'm gonna get this project done at some point. It'll be done. I'm just gonna briefly mention these. These are the TG-CRC projects that my PI Jean-Fei Liu is working on. So he's working with Carlos at UT Austin and Dr. Alper, whom I'm also working with on this project, deployment of plastics. So actually looking at the environmental real-world interactions with microbes and plastics, which should be pretty interesting to see. And we're doing an ALE in the lab to try to isolate some of those microbes that can use the plastics, not just the DOC. I'd like to thank Dr. Jean-Fei Liu in the Liu Lab, Dr. Alper, Ryan, and Keisha, and all of my funding and support. And thank you. Yeah. Yeah, that was the I think I understand the question. It was the DOC from our photoradiation. So we photoradiated the nurdles, they produced the DOC, and then we took about 2.3 milligrams per liter of that for each of our vials and inoculated those with some microbial consortia. So I think it was about 10 milliliters of our DOC from that photoradiation period, 10 milliliters of our natural microbial consortia, and then artificial seawater. Yeah. We don't know too much. Theoretically, these samples are like standard nurdles, so they shouldn't have too many additional additives or toxicants that are going to inhibit the plastic growth. Um, but that was just sort of like a general hypothesis, maybe because the growth was a little bit lower than the vials with just natural DOM. I haven't looked too much into it yet, and hopefully the LCMS data will actually give me some more understanding of those chemical properties, and maybe if there's anything that is typically inhibitory for plastic growth, and we'll see if it actually is the case. Um, Let me think. Hold on. So you're saying like the biofilm and then the like DOC as well, or yeah.
Would the biofilm be protecting, protecting the neurons from the light, or is the light breaking the neurons down?
Yeah, absolutely. Mhm. I think it probably depends on like where the nurdle was already at before that biofilm was formed on it, and also it's hard to completely apply this data to the environment because you have all these other factors of where the nurdle is in the water column. A lot of these are pretty dense, so they're going to be in the sediment and not actually getting that photoradiation. So this is like maximum photoradiation, best circumstance of the photodegradation that we're going to get, and obviously the biofilms are going to interfere. Any sort of mechanical degradation is going to take off that surface layer that has already been degraded by the photoradiation, and you have sort of a second starting place. So there's a lot of factors that are influencing it, and it would definitely, affect.
Okay. We have a 30-minute break, and then we'll meet back here at 10:30 with a talk about cyanobacteria and wind tidal flats.